Chapter 5 is all about the biomolecules that are found in every living thing, and that includes carbohydrates, lipids, proteins, and nucleic acids. Now, you'll notice that we skipped Chapter 4. Chapter 4 is about organic chemistry, and you don't need to read Chapter 4 closely, but I would like you to be familiar with these functional groups. There's no test. I won't quiz you on what's a carboxyl group or a phosphate group, but I am going to use these terms like you already know what they are. So you'll want to familiarize yourself with the functional groups on this chart from Chapter 4. So for Chapter 5, we're really getting into the first material that's going to be new and detailed and specific to biology. And so I've put together a study guide for you to fill out where you'll see each of the different biomolecules matched up with each of these different things that I want you to know about them. And so we're going to start with carbohydrates. So carbohydrates, what you're mostly familiar with for carbohydrates are simple and complex carbohydrates that are sources of energy for food. Okay? So when you think of table sugar, that's a carbohydrate, as well as complex carbohydrates like starch that's in pasta and bread and potatoes and stuff like that. Okay? So one of the functions that you're already really familiar with is their form of energy, either energy storage, for long term, or for a quick source of energy for our cells. The simplest form of carbohydrates are, in other words, the monomers of carbohydrates are called monosaccharides. And monosaccharides come in all kinds of different varieties, but the base formula for a monosaccharide is for each carbon, there are 2 hydrogens and 1 oxygen. And then what varies is how many different carbons are in that molecule. And so one way that carbohydrates are named are by how many carbons are there. So a pentose sugar has 5 carbons, whereas a hexose sugar has 6 carbons. We're going to see throughout this chapter that form fits function, and by changing the shape, you're going to change the function or how it's used or perceived in the body. And one example of this is if we arrange the molecule or the atoms in a molecule of glucose or fructose, they're going to be perceived differently by the body. So both glucose and fructose have the same chemical formula. They're both C6H12O6. But the position of different functional groups in the molecules make them perceived slightly differently in the body. So on glucose, the carbonyl group is terminal, whereas in fructose, it's branched off of this second carbon. So molecules that have the same chemical formula but have different shapes are called isomers. So when you look at these molecules, what about them makes you think that they'll be either hydrophobic or hydrophilic? Hopefully you noticed all of these hydroxyl groups, all these OH groups, which are a part of water. So just like with water, how the oxygen side is partially negative and the hydrogen sides are partially positive, okay? Same thing is true for the hydroxyl groups. The OH that's attached to something else, some carbon-containing molecule. Okay. The oxygen part is partially negative, and so it's going to be attracted to partially positive things like water molecules and form hydrogen bonds. So in your study guide, you can note that carbohydrates are hydrophilic. When you put sugars in water, they actually fold up. They form rings in water. So our linear glucose molecule actually folds over, and the carbonyl carbon actually reacts with this hydroxyl group and forms a ring structure. So now the oxygen from this hydroxyl group is inside of the ring. So more often than not, since in biology pretty much everything's happening in solution, happening in water, you're going to see carbohydrates drawn as hexagons. And so you should get familiar with what they look like. So go ahead and draw this out in your notes. I'm drawing a little bit of a shorthand. Each of these points are a place where there is a carbon atom, okay? And so carbon has 4 electrons in its valence shell. It has room for 4 covalent bonds. So this carbon's participating in one bond with the oxygen there, one bond with the next carbon over there, and then it has a hydrogen and a hydroxyl group coming off of it. Okay? So the same thing is true for this carbon, and for this carbon, and for this carbon, and for this carbon. Well, something different happens on this carbon. So this is a carbon, but this is a hexose sugar, and so the 6th carbon is actually up here, and it has a hydroxyl group coming off of it, but it also has 2 hydrogens coming off of it. Okay. So this is our glucose molecule that's been cyclized. I want to point out that we can give a name, or really a number, to each of the carbons in this sugar, which will help us keep track of where different chemical bonds form. So this first carbon that's attached to the oxygen is the 1' carbon. This is the 2', 3', 4', 5', and then the 6' carbon is sticking out up here. Okay. So that's just a little bit of detail that I want to point out that's going to become really important when we start talking about nucleic acids later in the class. So we can take these individual monosaccharides and start joining them together. So I'm going to draw another monosaccharide over here, another glucose molecule. And I'm going to leave off a lot of the other details. Okay. So these 2 sugar molecules can react with one another. The hydroxyl group from one reacts with the hydrogen from the hydroxyl group of the other and leaves and forms water. When 2 molecules react together and water leaves, it's called a dehydration reaction. It's sometimes also called a condensation reaction because water is condensed. So to join 2 sugars together, the reaction that does this is a dehydration reaction. My picture's getting crowded, but a dehydration reaction is what kind of chemical reaction joins these 2 sugars together. And when that happens, what we get is an oxygen joining them together. And then the hydrogens are still there. So the new bond that's formed is called a glycosidic bond. So the linkage between monosaccharides is called a glycosidic bond. And so if we have 2 sugars joined together, that's a disaccharide. If we have 3 or more sugars joined together, that's a polysaccharide. And polysaccharides are usually what you think of when you think of carbohydrates. Usually when we're talking about carbohydrates, we're talking about complex carbohydrates. Simple sugars are monosaccharides and disaccharides. So some disaccharides that you might be familiar with include lactose, which is the disaccharide found in milk, and sucrose, which is table sugar. So when we think about the functions in different kinds of carbohydrates, our monosaccharides function is to enter cellular respiration to provide energy for the cell. These complex carbohydrates, like starch and glycogen, are used for energy storage. So starch is used for energy storage in plants, and glycogen is used for energy storage in animals. That's how carbohydrates are stored in our muscles. So when you carbo load before a distance run, you're converting the energy in starch into energy that's in glycogen, so your muscle cells can use it during a race. Cellulose and chitin are also carbohydrates, but they have a different function. Their function is structure. So cellulose is the most abundant biomolecule in the world. It's the fibrous material found in plants that holds them up and supports their cell wall. Chitin is a structural carbohydrate that's found in arthropods, in insects. Their exoskeleton is made of chitin, and it's also found in fungi. So I want to look a little more closely at these polysaccharides. So both starch and glycogen and cellulose are all polymers of glucose. They're all just glucose joined end to end to end to end to end by a dehydration reaction forming a glycosidic bond. But they have slightly different structures based on the orientation of the glucose molecules to each other. So in starch, we're going to see that each of these sugars are actually flipped around so that the sugar kind of makes this twirling helix structure. And starch in this shape is digestible by animals. Glycogen, the sugars are also arranged in that same orientation, but it's branched, and that's the way that sugar is stored in our muscles. Cellulose, on the other hand, the sugar molecules are flipped in a different orientation that allows the lines to line up perfectly. So if we look and zoom in on starch and cellulose, okay, so again, both starch and cellulose are polymers of glucose, but they're hooked up in different directions. So if we look at starch, here's one glucose molecule, and the hydroxyl on the on the 2' carbon is going down, just like the oxygen on the glycosidic bond. So all of these are lined up in the same direction. Well, putting all these hydroxyl groups close together is actually energetically unfavorable. These hydroxyl groups want to get far away from each other. So the molecule twists up into this helix shape. On cellulose, on the other hand, each glucose molecule is flipped in the opposite orientation. So here the hydroxyl group on the 2' carbon is down. On this one, it's flipped up. It's flipped down, it's flipped up. And in this orientation, the hydroxyl groups are already far away from each other. Okay? And so what that allows the cell to do is to line those molecules up straight across, and then they can hydrogen bond with each other and make these really rigid fibers that are not digestible by us. And I apologize, my special characters here got messed up when I switched from a PC to a Mac. But this first example in starch is an alpha-1,4 linkage. And an alpha-1,4 linkage is digestible by animals. Cellulose has a beta-1,4 linkage. And in this orientation, we don't have enzymes that can break that beta-1,4 linkage. Okay? So no animals can digest cellulose. That includes cows and termites. But there are prokaryotes that can break that beta-1,4 linkage. So herbivores that are digesting cellulose and termites, the way they're doing it is they're using their microbes to break down those beta-1,4 linkages. All right, so looking back at our study guide, one detail we left out here is that the polymers of carbohydrates are called polysaccharides. So starch, glycogen, cellulose, and chitin are all examples of polysaccharides. I wanted to make one more note about chitin here. So chitin is also pretty much just a polymer of glucose, except for on the 4' carbon, there's actually a nitrogen-containing group, an amine group. And we're going to see that that functional group is also found in proteins in amino acids. And so it's that amine group that kind of contributes to that savory umami flavor in mushrooms that kind of reminds you of meat, even though it's not really meat. So for our carbohydrates, the monomers or monosaccharides, our favorite example is glucose, but others include fructose and galactose. Our polymers are called polysaccharides, and our examples of polysaccharides include starch, which is the energy storage molecule in plants, glycogen, which is the energy storage molecule in animals, cellulose, which is a structural carbohydrate in plants, and chitin, which is a structural carbohydrate in arthropods and fungi. The monosaccharides are joined together by a dehydration reaction, which leads to the formation of a glycosidic bond. And they're very hydrophilic because of all those hydroxyl groups. So in addition to knowing the words, you guys should also be able to recognize the structural formula. So when you see this molecule, you should be able to recognize that that is a monosaccharide. So be able to visually recognize them as well as know the vocabulary words.